Okay, second time recording because I was just blathering on too much. I'm going to try to stick to the essentials here. So I'm introducing RNAi. I'm introducing it here not because we're going to be talking about it in detail in this section, but because in many organisms, this is one of two main techniques that are used to directly target particular RNA to see what a loss of function phenotype looks like. Loss of function meaning that no functional gene product is being produced. And in this particular case, we are talking about mainly the production of a functional protein. So the way you do it, you have to know your gene and you have to know its sequence. I'll get rid of this stuff that I put in the last time I was trying to do this. Let's do purple. So you have to know the sequence of the gene that you're targeting. You need to have some idea what it's doing. So we think this particular gene is probably needed for the production of P granules in a nematode embryo. And so what we do is we take that gene, we find out what the RNA, the messenger RNA sequence is, and then we don't have to make a whole double-stranded RNA of the entire one. Usually, around 100 to maybe 300 or 400 nucleotides is enough. It can even be shorter than that, but in my experience, this works really well, at least in flies. And so you want to make your RNAs that are double-stranded. Of course, you remember RNAs are normally single-stranded. This technique is based on a mechanism that cells, eukaryotes, appear to have as a protection against certain kinds of pathogens, mostly viruses, RNA viruses. Some of them, when they infect cells, have a double-stranded RNA stage. And most of the time in cells we don't normally have double-stranded RNA, so that's a cue for the cell that a pathogen is trying to invade. And so there's a mechanism in the cell for targeting that double-stranded RNA and for targeting the RNA that matches it, even if it's single-stranded. So you make this double-stranded RNA and you introduce it into the organism or the tissue or cells, however you're planning to do it. It's done a lot in tissue culture, for example, cultured cells. And I won't talk about how you make the constructs or any of that because that is for other courses. But if you're interested, you can always ask. I can tell you a little bit about that. The double-stranded RNA, as I said, it triggers a response and in the case of us targeting a gene that's actually in the organism itself, it will target the normal messenger RNA for that gene as well. And not only will it destroy the double-stranded RNA, but it will then destroy the normal messenger RNA. And this gives us a loss of function phenotype, no functional protein. Now, what's really interesting, and I won't say too much about this because I want to try to keep this really short, but there are a number of organisms that have co-opted this mechanism of protection, and they've modified a little bit to use in regulating some of their own developmental genes. And we might touch on a few of these processes in subsequent chapters. So I mention it here so you're prepared when you hear about it later. So this is how it works. You manufacture your double-stranded RNA, and as I said, it doesn't have to be full length. It can be on the short side. There are some organisms that use very tiny RNAs called microRNAs to do this kind of regulation. It gets into the cells. In worms, it's super easy. There's a multitude of ways to get it in, but you can even just feed them bacteria that have a construct that's producing double-strand RNA of your cell, your gene of interest. And if they eat the bacteria, they wind up with the double-stranded RNA inside. And they take enough of that in to produce a phenotype, which is super cool because it's a lot more complicated in most other organisms. Some of you know that CRISPR is used in place of RNAi in some cases. But RNAi works in all the eukaryotes that have ever been tested for it, and it is actually quite effective still. Let me just remove my mistakes from the previous recording. Okay, so basically we introduce that into the cell. And then a protein called dicer that's there as part of the surveillance system will recognize, oh, double-stranded RNA, that's not good, that's probably a virus. And so there are then 4 molecules, a homotetramer of the dicer protein that bind it. And it binds us all over the place. It doesn't matter where it is. It'll bind anywhere along here. But the size of the proteins binding here defines where they cut, and they do cut the double-stranded RNA. And so what you end up with is little pieces, and there are approximately 22 nucleotides in length, 21 to 24. Different organisms they're slightly different, but they're little tiny pieces. And if you've put in a 200 base pair piece, it's going to be chopped all over because the proteins don't bind just the molecule one time. There'll be some more over here and some more at the other end, and you'll end up with all these little short interfering RNAs, they're called. So these short interfering RNAs are bound to the RISC complex. And the RISC complex is a group of several different proteins. And the main things that are done is they bind these little double-stranded RNAs, but one of the strands is discarded, and just a single strand with its ability to do complementary base pairing is retained. And that's referred to as a guide RNA. And the guide RNA is the one that now is going to be used by this complex. And if there's any messenger RNAs that have complementary sequence to this 22 nucleotides, then the protein is going to bind that RNA and it's going to chop it. And that is how it prevents the expression of the gene. It hasn't affected the gene itself, but what it has done is prevented the function of producing the protein. So here's our gene. It's being transcribed inside the cell. Here's the messenger RNA. Out it comes, oops, not with an eraser. Out it comes to the nucleus, or out of the nucleus into the cytoplasm. And here is the RISC complex with this guide RNA, and the guide RNA goes, oh, okay, I can bind to you with complementary base pairing because I have the complementary sequence. And then the RISC complex goes, okay, out you go, and it chops to either side of that to chop and it can bind, another complex can bind sort of somewhere else on the same one and chop it into small pieces. And so there hasn't been a retained ribosome binding site, or if there is, it's right at one end, and then 2 or 3 amino acids can be produced in a string, and that's it, because the RNA has been chopped to pieces. And so basically that's how you target the gene. And so in our example with the P granules, so if we thought this gene was required for P granule formation, we would feed the worms the double-stranded RNA or bacteria that is expressing it, and then we would see if the embryos from these hermaphrodites had normal P granules, and that would tell us if we were correct about our prediction or not. Maybe the gene is actually doing something, but it's not specifically about forming the P granules, it's about something else. Maybe they form properly, but they don't localize properly, or something like that. Okay, I think that's enough. Let me know if you have any questions, and we will see you next time.